So I'm going to talk today about Harmonizome, but I'm also going to mention some other tools and resources that are related. And let me get started. So I'm going to start from the many years ago. I joined Mount Sinai as a PhD student 23 years ago in 2002. And I was not directly, that was not my first career. So before that, I was a software engineer at some mid-sized company doing programming, and I decided to switch fields and get my PhD in biomedical sciences. So I had some computer science background, and for my thesis, I chose this project that tried to create a cell signaling network that connects, extracellular ligands to different cellular machineries. And the idea was that all, cell signaling pathways actually connected and they all forming this large network. And potentially, if we understand how this network is configured, we can potentially understand how a cell sort of like is regulated and function. And we came up with this diagram that you can see. It's very messy. And honestly, it did not help us to really understand how the cell works just by looking at this diagram, even though here all of the interactions are direct physical interactions and the arrows are mostly, signed and directed arrows. So it's like whether it's activation or inhibition, you can still really cannot tell how it works. So just by looking at that. So the paper, nevertheless, was published in Science, which was very helpful for my career. It sort of like helped me to get a lab, and um, it was one of the breakthrough of the year in 2005. And that was my thesis project paper. And for that project, also what I tried to do, I tried to read a lot of papers that talks about cell signaling. And then from those papers, I manually extracted those interactions between those proteins. And my idea was like, okay, maybe if I have it all in my brain and all that information about those components, those cell signaling molecules that are mostly proteins, that how they're all connected, I will be able to figure out how the cell works, and it will be all in my mind. So that project also required me to read, I think it was over 1,500 articles, like, regular sort of quote unquote, back then it was like a regular JBC type of articles where people just try to figure out how this kinase phosphorylates some targets and what effect it has on cells. So anyway, at the same time that that paper was published, there was also News and Views article that tried to place the paper in the frame of other papers. And at that time, a lot of people were interested in dynamical modeling of biological networks, specifically cellular networks. And the field sort of like abandoned that approach for most part, just because we realized that it's so complex. But if you think about it, if you could potentially have very accurate simulations of few cellular components. For example, you can simulate how two proteins potentially bind to each other and using molecular dynamics type of simulations. And then if you go into a larger circuit, you can use ordinary differential equations. And then if you get larger, you can use Boolean, that type of dynamics. And then our approach was even more abstract and even more simplified. And we tried to simulate this network, but without much success. And then since then, my lab stayed in the even more abstract place of trying to understand, molecular biology, molecular systems biology. So the first project that we did as an independent lab, and it was published in 2010, was this project called KIA, which is kinase enrichment analysis. And here, what basically I realized is that instead of reading those articles myself and try to extract those articles, those interactions from publications, there are already a bunch of databases that had that information for kinase substrate interactions. So we took the data from those 6 databases and we integrated them into one database. And then we built this tool that allow you to come in with your differentially phosphorylated proteins from phosphoproteomics or differentially abundant proteins from proteomics. And then you can submit them to this tool, and then it can give you a ranked list of kinases that are potentially overrepresented with substrates, and then prioritize those kinases to tell you which kinases are likely regulating those proteins. And this was app that came with the paper that gave you the ability to just cut and paste your gene set or your protein set and get the kinases. Very similarly, sort of like around the same time, we also realized that instead of reading a lot of papers and extracting those binary interactions, there is some papers that are specifically telling you how transcription factors regulate many target genes. So this was done with ChIP-seq and ChIP-chip and other chip, like there was a ChIP-PET type of method to determine where transcription factors bind on the genome. So we collected those papers, 212 papers at the time had transcription factors, single transcription factors. Sometimes they did 2 or 3 transcription factors because those experiments are pretty hard to do. And then we aggregated all of this data. So we had to normalize the gene IDs and the transcription factor names, but it wasn't too hard. And then we created this database of transcription factor targets, and we called it CHIA. And then CHIA also came with an app where you put in your genes. Here, those are differentially expressed genes. And then you can get the transcription factors that are most likely regulating those target genes. And this was featured on CNET News back in 2010. So maybe going forward a couple of years, we realized that this can be done for a lot of other types of data. It doesn't have to be just ChIP and KIA. Actually, there's so many other databases that can be, I guess, normalized or abstracted to various data structures, like gene set libraries. But also you can make bipartite graphs from those resources. You can build networks that are connecting either the genes or the entities from those resources based on their similarity, and other types of data structures that I will also discuss. And then we found that there are different categories of this type of data. So there's many databases and different types of categories of data that can be normalized. And here is a summary of them. So in this review article that we published in 2014, we listed those 7 different types of data that can be converted into those gene set libraries or those networks, what people called back then functional association networks, and today they're called knowledge graphs. So this is I'm going to elaborate on this. So the same, in the same review article, the same concept. Here, we show how those different data sets, once you convert them into this format, you can start making connections based on their metadata or their description of the gene sets. And specifically, you can connect, for example, cancer cell lines to drugs. And you can connect drugs to side effects. And now all of a sudden you can connect side effects to cancer cell lines, and then cancer cell lines to tumors from patients, and etc. So all of those connections can start help you with doing data harmonization. And at that time, it was sort of like a theory that you could do all this integration. But what we've done also, and we had specifically a very dedicated postdoc that went to those resources, like to those many databases that I mentioned, and systematically wrote scripts to convert the data into this particular format. And then here you can see that at a high level, you can really abstract this knowledge into this high-level concepts that are really divided into 5 different types. So you have those genes and proteins at the center, but then you can have diseases, phenotypes, and adverse events that are potentially all clustered in this one type of category. You have gene modules and pathways that are can be also considered another types of set. You have drugs and small molecules, and then the cells and the tissues are another set. And everything can be connected to everything, and many biological projects, so biomedical research articles and databases can fit on one of those edges, making those connections. And if you want to expand it a little bit more, you can include patients, you can include assays and experiments, and pathogens like viruses, bacteria, and other pathogens. And everything, again, is connected to everything. So this really enable us, after writing all of those scripts, to create those 2 highly impactful apps, Enricher and Harmonizome, both published around the same time. And then we kept on upgrading them and updating them. So the community of users grew over time. So people realized, hey, this is really useful. And I'm going to talk today mostly about Harmonizome, but I'm going to mention some of those other new apps that are related to it. And sort of like what I want to bring up is this concept that I just described. So Harmonizome currently has over 106 million associations between 60,000 genes and 500,000 attributes that, for example, could be a pathway or a phenotype or a kinase or a transcription factor. And those are extracted from 84 resources. And some of those resources, for example, GTEx or links, may have multiple data sets that are generated from those resources. So the total is 164 data sets. So those are, again, divided into those categories that I mentioned. And then there's also subcategories and metadata that is collected to describe each library or data set, but also specifically each set of genes that are just associated with some type of a term. The real benefit of doing that for Harmonizome was the gene pages. So Harmonizome has gene landing pages that provide the information about this, all of those associations. But one of the things that we added to Harmonizome 3.0 is those automatically generated description about each gene. So to do this, we searched for the gene name on PubMed. We took the top 50 articles that had the most citations that mentioned that gene. And then we gave those to ChatGPT or GPT-4, and then asked it to summarize all of those 50 papers into one short description of the gene with citations. And it did a really great job for that. So as I will talk later, you have to be very careful of how you're using LLMs in your bioinformatics applications. But here, for this particular task, it's doing a really great job. And you can see that here you get a short paragraph that is superior to the paragraph that you get from NCBI. So this is the description of the gene at the top is the original description for this gene from NCBI. Below is this automatically generated description of this gene based on this method. So this is a cool feature that was added to Harmonizome 3 that was not there before. And here I'm showing specifically the gene page for CD64 that currently is my favorite gene, and I will explain to you why. So the next, once you scroll down the gene page, you can see those associations, and there are over like 160 of those foldable entries. And here you can see all the A's. So it's just the first 5, and you can expand it and see, for example, which cell lines are sensitive to the knockout of this gene, and which one are like potentially increased proliferation and decreased proliferation, and based on Achilles. So I mentioned that gene, like why do we care about this particular gene? Why it's now my favorite gene? And this is because we found that genes to be highly expressed in triple negative breast cancer tumors based on analysis of the CPTAC triple negative breast cancer using our tool Target Ranger. So what Target Ranger is doing, it takes the expression level of each patient's tumor, and instead of comparing it to adjacent normal or to other tumors from like a cohort, we compare the expression of the tumor to many normal cell types and tissues from 3 atlases. So this is Tabula sapiens, GTEx, and Arches 4. And then we look for self-surface gene that gives rise to proteins that are self-surface that are highly expressed in the tumor and lowly expressed across all of those normal tissues. And we found 58 of those proteins or those genes that are uniquely highly expressed in those 28 breast cancer patient tumors. And then what we did, we looked at depth map and for those 58 targets, and so which one of those are the most sensitive to knockout across all of the triple negative breast cancer tumor cell lines in depth map. And we found that this particular gene is highly sensitive to the, like if you knock out in triple negative breast cancer across many of those cell lines, this gene CD64 is a great target potentially for that subtype. So that's why we're interested in it. And this sort of like advertise a little bit about Target Ranger, which is a different app and tool, and Harmonizome. Another important feature of Harmonizome is the dataset pages. And every dataset out of those 160 datasets is served on this dataset page with some a description and references. It tells you how many genes, how many terms are there, and how many associations. And also you can get the ETL script that converts the data, the raw data from the source into the format that is available on Harmonizome. And Harmonizome provides multiple types of files. So here you can see there is like gene set libraries. You can get the app genes. You can get just the attribute list. You can get the gene list. You can get the other, you can get an attribute table that you can use for machine learning and some other things like that. For example, knowledge graph serialization for ingesting into a knowledge graph. This landing page is for drug perturbation that we collected from this resource called RamaGeo. So RamaGeo is another app or resource that we produce that I'm going to talk about. Once you scroll down the dataset page, you also get those visualizations of the data in the dataset. And the UMAP is a visualization of all of the gene sets in the datasets based on their similarity. So you can see that those drug perturbations cluster into nice patches of gene sets that potentially have commonalities in their response to those drugs. So we have 2,867 sets of up and down genes, about 300 drugs, and some of those drugs have multiple entries for multiple, and all of those are extracted from geo studies. So RamaGeo is a relatively new app that was published last year. And for RamaGeo, what you can do, you can come in with your gene set, typically a differentially expressed gene set, and then you can search through 178,000 gene sets that we automatically computed by comparing groups of samples from Geo. So this is a search engine for gene sets that are automatically generated from RNA-seq studies in Geo. So once you have all of this data in this format of gene sets, you can start crossing data sets. So here, Harmonizome 3 has this new feature that's called dataset crossing. And here you can select from all of those data sets two pairs of sets. For example, here I selected cell marker gene cell type association. So those are genes that are specifically used to annotate cell types and typically used for cell type annotations for single cell RNA-seq. And then this GeneNet, which is a famous database that associate genes with diseases. So you can see that, for example, this B cell is also associated, the genes that defined this particular B cell are also associated with a B cell malignancy low grade. And then you can click on this generate hypothesis with GPT to sort of like understand the overlap. This concept is also implemented in this other app that I want to mention that we published this year called GeneSet Cart. And this was one of our efforts for the CFDE. And gene set card was developed so you can manage your gene sets. As you do analyses, you want to make sure that you can go back to various analyses that you've done, and you want to have sort of like a bank of the sets that you are working with, so you don't have to re-upload them all the time over and over. And within gene set card, there's this feature where you can cross gene sets from different sources. And here I'm just going to show an example of crossing gene sets from GTEx and Motopac. So using the GTEx database, we created aging signatures by comparing the young groups of tissues. So for those of you who may not sure about like what GTEx is about, it has about 800 postmortem donors, tissues from donors, profiled with genomics and transcriptomics, and they took the 54 tissues from those donors and profile those with transcriptomics. So you have different ages and tissues from those patients, and then you can do various analysis with this data. And one thing that we've done, we compare the young versus old across all of those tissues. And then Motopac recently produced data from tissues of rats before and after giving them exercise regimens of aerobic exercise of 2 or 4 weeks. And then they measure their changes in what happens to their tissues before and after exercise. So now you can take those gene sets that are up and down across all of those tissues and compare them. And the two sets that have the most overlap come from blood, from GTEx, both, and also blood in the female rats. And the genes that are upregulated due to aging are highly overlapping with the genes that are downregulated in blood towards exercise. So those are interesting genes that potentially we want to downregulate in order to potentially mitigate some of the effects of aging and mimic the effects of exercise. So again, we have this button here that you can press on to get hypothesis of what's happening. And specifically to generate that hypothesis, we give some background about each of those gene sets and data sets. We give the genes themselves, as well as the results from enricher. And we ask the LLM to compose an abstract based on all that information. So we very much restricted to this domain. And it shows that really what stands out is blood clotting and cholesterol. So the overlapping genes are related to those two pathways. So it helps you very quickly to see the cause of the overlap. So one of the things that Harmonizome is really potentially enabling is the ability to take all of this process data and do machine learning. And you can be creative of what you can do. But one of the obvious things is predicting gene function. So what you can do, you can take those attribute tables from those various resources. For example, you can take a table from transcriptomics, either tissue expression or perturbation, and then you can take proteomics data, and you can take literature-based data about pathways, and you can concatenate all of those attributes to build attribute table. And then if, for example, if you want to predict the phenotype of a knockout mice, of a gene that has not been knocked out in mice, you can build this machine learning function using like libraries like scikit-learn or TensorFlow. You put all this data and then you can get predictions of for those genes that have not been knocked out in the mouse. If you want to predict a specific phenotype. So for example, for in mice, there is some phenotypes that are beneficial. For example, low cholesterol, or so some mice that have a knockout of a specific genes, they have lower cholesterol than other mice. Or some mice, they live longer. So you may want to find additional genes that are predicted to have this phenotype, this positive phenotype. So this can be done with machine learning, like sort of like traditional machine learning. But what we've done recently is that we tried other methods to predict gene function, and we are using semi-supervised or unsupervised approach. And we're using those foundation models or deep learning approaches to do it. And specifically, we take all of those gene sets and we give them to this gene set foundation model, and we give only like a partial of this set. And then we ask the model to complete or fill in the missing part of the gene set. And then that gives you a model that can potentially be used to make predictions about gene function, as well as to do other things with it. Over the years, we tried also standard machine learning approaches and other methods to predict gene function, like based on just co-expression data. And other people have also tried to use similar approaches to predict gene function based on protein-protein interaction network position, based on co-expression, based on literature and the description of those genes. And here we show that this new model that we just published 2 weeks ago is superior for predicting gene function, both when trying to predict data that is not from literature, as well as literature-based data. So this is published in bioRxiv, and it's called Gene Set Foundation Model. So this gene set foundation model is trained, it's not trained on the Harmonizome gene sets. It's trained on these other gene sets that are created for this other app called RamaGene. So RamaGene is similar to RamaGeo in a sense that it's a search engine that you can come in with your genes, and then you get results based on that match those gene sets. But with RamaGene, what we've done is we went to a PubMed Central. The PubMed Central currently has almost 7 million articles that you can access programmatically. You can download those articles, you can scan them. They are publicly available for data mining. And from those articles, we specifically went to the supporting materials. Those supporting materials of articles typically are not searchable, and they are hard to extract knowledge from. They're typically in PDF files and Excel spreadsheets, and they're not standardized. So in those supporting tables and spreadsheets and PDF files, we try to look for gene sets. And we scan for those columns in those spreadsheets, and we try to see if we can extract or find human or mouse gene sets from those tables. And then if we found them, we added them to the database. So out of the almost 7 million PubMed Central articles, we are able to extract 881,000 gene sets, and only 100,064 articles had some gene sets in their supporting materials. And the nice thing about this, it's actually these numbers get updated weekly. So this is a huge collection of gene sets. And what we've done with those gene sets, we fed them into this foundation model, and then that foundation model can be used for gene function prediction and potentially other downstream tasks. So this is what happens if you submit the example gene set to RamaGeo. You get matching gene sets based on the overlap that is calculated using the Fisher exact test. And here you can start examining the gene sets that show high overlap. The reason it's called RamaGeo is because you get a lot of results. So you have to rummage through those results to see if you get things that are useful for your research. The last feature that I want to mention about the new Harmonizome 3.0 is a chatbot. And this is something that we try to add to multiple different apps that we've developed, and other people are trying to do it now. And the idea is that this chatbot uses LLM in the background, but also has access to APIs. So we gave the chatbot all of the Harmonizome APIs, and then you can see that it has a very structured answers, and it helps you to get answers from Harmonizome by a combination of text and the APIs that are in the background. For example, if you ask it a question, like very basic questions like, what can you tell me about stat3? It will just give you the page of stat3 and potentially give you the text from the that I showed you before that is automatically generated from the top 50 abstracts. One of the other things that it has, it also doesn't really only answer with text. It can produce tables and it can also produce figures, but overall it doesn't work that well. So it still requires a lot of work to get it all the way right. So now I'm just going to summarize the entire presentation. So first, the main point is that converting omics datasets into those simplified data structures, such as gene set libraries, also knowledge graph or attribute tables, can really facilitate data integration, data reuse, and knowledge discovery. And also, once you start triangulating evidence for multiple resources, you can really narrow down your hypotheses and really find things that are really worth it to test experimentally. So once you get this, it's sort of like investigating a crime scene and getting evidence from different sources to prove a case. Once you have enough evidence for multiple resources, the answer is clear. And now we can just need to prove it experimentally and show that this is a new discovery. You can also prioritize your hypotheses to decide what would be the most impactful prediction, what would be the best. So once you get those multiple high quality hypotheses, you can really prioritize them once you have them in this order of likelihood to succeed, as well as impact and novelty. So gene function predictions can really fill our knowledge about the function and role of genes in multiple contexts like disease, pathways, and other things. And this is because right now we are in a great state in the field that we have partial information. So we have a lot of data and we know a lot about a lot of genes. And then we also have a lot of genes that we don't know anything about or we know very little about. So ML and DL methods can really help us to fill in that gap can make the pictures clearer. And then this data, once it's processed and harmonized, can be used to build those foundation models that can be very powerful for multiple tasks. So I only talked about gene function predictions, but you can do enrichment analysis, you can do protein-protein interactions predictions, and some other stuff that I can talk about later. And then one of the other things is that when you're using those LLMs to enhance your bioinformatics platforms, you have to be very careful, but you can be creative and they can be useful. Their adaptation is going to be a challenge because people don't really trust them, but they're coming and they're going to be here for the long. So I just want to mention that I also spoke about that a little bit. And finally, I would like to thank all the members of the lab. This is all of their work. Some of the instrumental people for Harmonizome are Daniel Clark and Ido Diamant, who has done a tremendous job. And several of the projects that I mentioned are led by Daniel, who's been in the lab for now almost 7 years, and it's been like a super productive and creative contributor. And also, I would like to thank the NIH for their funding. And now we can have some discussion and questions if anyone has any questions and would like to discuss anything. Let me stop.
All right, great. Leon, I think you have your hand up.
Yeah, I have a question. Oh, thank you so much for the great talk. I think it's very productive job. I didn't realize that we actually use your tool also in the team, like in HR also. So my question actually refer to the Chat GPT. I think it's quite interesting that you actually use Chat GPT to summarize those like 50 most cited abstract and then also kind of summarize that in one paragraph. I was just wondering because we also do something similar, but we just realized, sometimes ChatGPT just make up the citation, So in terms of like how did you actually kind of validate the output from ChatGPT? Could you talk a little bit more about that? And also, how fast to use ChatGPT to summarize, a lot of abstract, right? I think for your case here, you maybe just pick up only 50 abstract, but if you are going to do um, for large data set, and then how quick or what about the running time? Can you share a little bit with us?
Sure. So obviously I did not do it, but so my answers might be not 100% accurate, but I think it took about $700 to do this for all of the human genes. And it also took about 24 hours to run.
Mm.
And it had to be cleaned up. So we know the references because we give it the references. And we had to sometime, we did not use the references that it put directly, but we actually replaced. We knew the one that the ChatGPT thought he was putting in there, but it had to be replaced with the original ones. So you can have to do some cleanup afterwards.
Mm.
And also the prompt engineering is very important. So you have to know exactly what to tell it and how to make it not, the temperature has to be adjusted. The prompting had to be done very well. So yeah, this is you have to play with it.
Got it. Great. Thank you for the answer.
All right.
Do you have other questions? All right, we have one here for gene function prediction. Does this mean that you can predict some gene functions which have never been discovered in any literature, or is it more about searching and finding out the gene functions from the existing literature?
Yeah, it's a great question. And the answer is absolutely, we can find gene functions that have never been discovered in any literature because we're using omics sources. So we use the co-expression, but not just co-expression. We use some proteomics, and some other sources. And with the GeneSet Foundation model, it's a mixture of literature and data from omics and other studies. So overall, definitely we can predict novel functions that for genes that have never been studied, because a lot of the data come from experiments.
So follow-up question on that, how do you validate that the prediction is correct if it's a new result?
Yes, so the way we do the validation, we take annotated gene sets. For example, if we do GWAS catalog, so those genes are associated, or let's say another one that is not literature-based, it's the mouse phenotypes for knockout genes. And then we take, let's say, an annotated set, and then we ask how well the model ranks the known genes for that phenotype. And we can basically do a leave one out, or we can hide part of the set and see how it recovers the other part. But you can do it using those gene set libraries. So this is what I showed in one of those slides. You do like a area under the curve for the ability of the model to rank and prioritize the known genes, given, let's say, part of the knowledge.
Maybe I can ask a follow-up on that. So I'm wondering if you could elaborate on, so I know you tried several different models for how to build this foundation model. And I'm curious about if you have any insight into why the one you chose worked better than the others. Like, is it picking up some latent structure that is like some gene functions, or just if you've thought about that?
Yes, so one of the things that with gene sets that is different from LLMs is that the order doesn't matter, like the order of the genes. So initially we thought, oh, we're just gonna use a transformer model, but the transformer models did not work that well because they are expecting ordering. So they are designed for language or things that are sequential. So gene sets are not sequential. So the even simpler models like autoencoders are working better. And then the reason one of those, I think we tried maybe 14 different models, different architectures. And then the one that worked, I honestly don't know why that one worked the best, but the idea is that it captures the enough of the space to generalize and also understand the nuance of the different types of data that it sees. So genes have different context in different sets, and sets have different functions, and they come from different sources. So that space needs to be captured, but also generalized as much as possible. So it's like a balance between capturing the space and generalizing at the same time.
Okay. All right. Another question. Hi, Michelle. For gene function, can you specify the tissue you're interested in for the function? So I think this question is, can you predict function specific to a tissue, given that co-expression can be tissue specific or cell type specific?
Yeah, so we addressed this a little bit in prior publications. So the short answer is not really with GSFM, but we can generate a co-expression network for a specific tissue automatically with tools like Arches 4, and then we can predict gene function with that co-expression network that is tissue specific. One of the things that you could add to GSFM, you can add like a text module that goes directly with the gene set, and then you can potentially ask that question. But right now it's not there yet. So there are ways to do it, but you know, we sort of like know how to do it, but I haven't done it completely yet.
Okay. Other questions? All right, I'm gonna ask, maybe switching gears a little bit. I'm curious. So you mentioned kind of knowledge graphs and how a lot of the work you're doing is obviously related to knowledge graphs, but I think, you know, you've definitely taken a different approach, I think, than a lot of people who are trying to assemble these heterogeneous and curated knowledge graphs. And, you know, and a lot of, especially for like rare disease diagnosis, you see a lot of the motivations for these knowledge graphs to be building these corpuses that you can then use to help predict genes involved in rare disease. And I'm curious, I mean, nothing specific, but more just your thoughts on like the field of knowledge graph construction and whether you see it as kind of the same thing that you're doing just via different lens, or whether you think that it has strengths or weaknesses compared to the approach your group has taken, and why you favor kind of where you've arrived at relative to some of the more like curation and ontology and like knowledge graph construction efforts.
Yeah, I think knowledge graphs are limited because of the explosion of the space of interaction. So networks and knowledge graphs are sort of like the same thing, and they need to be really sparse to be useful. But in reality, the data is too dense. So if you have thousands of edges between every node and every, you know, and you have like millions and billions of edges, it's becoming unruly. And then it's hard to um, sort of like make useful sense of it. And it's harder to understand what's there. But it's not completely useless. Like I'm not trying to say that it's not, but from what we've faced, it's becoming a bottleneck to represent the data in that format if the data is too dense. If you have, for example, for transcription factors, you have for every transcription factor, you have about 1,000 to 2,000 targets. So representing that data in a knowledge graph is too dense and there are better ways to deal with that data. For things like gene disease associations or pathways, maybe knowledge graphs are good. So I'm thinking, I think it's sort of a convenient way for people that don't really want to get their hands dirty with the code. It's a convenient way to load it into those existing knowledge graph databases but that accept that this edge lists, and so it just became easier to work with those types of data. The other thing that happened, you know, that happened already in the last, I guess, 30 years, is that people are obsessed with ontologies and annotations and making sure that the terms that describes those things are all harmonized. And we found that that's also a time sink a little bit. So if you are so worried about, you know, aligning those ontologies, it's definitely an important effort, but it can be a time sink if it's done for the sake of just harmonizing the data before you understand actually what you're going to do with it. So if you just do ontologies for the sake of ontologies, or metadata harmonization for the sake of metadata harmonization without a goal and without a clear path for discovery, you can waste a lot of time and a lot of money. So I'm not saying it's bad, but I'm just saying it's been overdone in the last 30 years. So yes, we are doing a little bit of it, but we try not to fall into this pit of uselessness or unproductivity by just focusing on metadata curation type of thing.
Okay. All right. Last Any last questions? I'll ask one final to close this out. Like, if you had to suggest one resource that, I don't know, somebody at Children's who's looking at a case that you've built should, and has a set of variant list or something, what do you think people should be using amongst your lab resources that your lab has created?
I don't really understand what you mean. Like if you have a list of
Yeah,
variants.
so you sequence a case and you have like 50 variants that are candidates. What do you think of all the tools you've built you think that people should take a look at to help with like variant interpretation?
Yeah, I think you want to convert them to genes and then submit them to enricher.
Submit them to Enricher.
Yeah.
Yeah. So Enricher, yes, that's everyone should be checking that out if they have not. Okay, so thank you, Avi. That was a great talk. Really, really interesting. And again, I encourage everyone to check out Enricher and also all the other resources and the CFDE if you have not heard of that. And thanks again to Avi, and thank you all for attending, and we'll see you soon.
All right. Bye. Thank you so much.
Thank you both. Have a great day.
Take care.